Well, now that you know how DNA is structured, you're ready for chapter 2.7 on how it works. This is going to cover DNA replication, transcription, and translation. So first we're going to talk about DNA replication, and replication just means to copy. Now, this is a process that cells don't go through just because they want to, because they're bored, because there's nothing on TV. The reason that they're doing this is because they're getting ready to go through mitosis. And so one of the things that the cell has to do before it can go through mitosis is to make a whole entire copy of all of the DNA so that each of the new daughter cells has its own complete copy. Now, this process is going to happen during interphase, specifically the S phase of interphase. Okay, and in eukaryotes, we're still going to be in the nucleus. So remember that in eukaryotic cells, we still have a nuclear membrane during interphase, and that's when a new copy of the DNA is going to be synthesized. So we're here in the nucleus. Now, there are two basic things that we're going to need. We're going to need enzymes, okay? Always the answer to an IB biology question is probably enzymes or ATP. So in this case, we're going to need enzymes, and we're going to need free nucleotides, okay? So that's these things. So remember, nucleotides are a 5-carbon sugar, a phosphate group, and a nitrogenous base. And we've got to have a bunch of those building blocks in order to synthesize a new strand. To make a new strand, we're going to need a template strand on which we're going to build the new strand, okay? So we got to make a template strand. How do we do that? Well, we take the double strand of DNA and we got to split it. Okay, that of course is done by an enzyme. In this case, it is helicase. So remember, the ending ASE tells us that it's an enzyme usually. And helix, okay, reminds me of that double helix shape here. Okay, so what helicase is doing is it's breaking these hydrogen bonds that generally hold the two strands together. And when it breaks those hydrogen bonds, it separates the two strands so that they can be used as a template to build a new strand. So after those two strands are separated, what that's going to do is it's going to leave us exposed strands here. Okay, now present in the nucleus again are a bunch of these free nucleotides, and those free nucleotides are going to be added to this exposed strand using the rules of complementary base pairing. So wherever there's an exposed G, then a free C is going to bind there, okay? Wherever there's an exposed A, okay, we're going to attach a free T molecule. So that complementary base pairing is really important in DNA replication. So what's interesting about this process is that to actually add a complementary base pair to an exposed strand takes nothing. Remember, they're held together by hydrogen bonding, by an attraction. So that part just kind of naturally happens. What still needs to take place, though, is you have to form that covalent bond, okay, between the sugar and phosphate groups, okay? So what I'm trying to say here is that this part, okay, the actual bases sticking together happens naturally, hydrogen bonding, an attraction between a positive and negative. But to bind the phosphate and sugar backbone, remember, to make this part, okay, we're going to need a enzyme, okay? And the enzyme that bonds those together is called DNA polymerase. So an easy way to remember that, again, ASE is that enzyme ending, polymer making a large molecule, okay? And in this case, the large molecule we're making is DNA. In real time, this process is wicked fast, okay? Faster than I can possibly show you. And the reason why that process is able to happen so quickly is, again, through this complementary base pairing. When it comes to DNA, if you're asked a question and you don't know what the answer is, I would start writing about enzymes and I would start writing about complementary base pairing. Okay, those are the general directions of all the right answers when it comes to DNA replication. Okay, so again, that process is super quick because there's not a whole lot of thought involved. Okay, G can only pair with C, A can only pair with T, and then we just get an enzyme to form the covalent bonds between the sugar and phosphate groups. Okay, so to kind of put that all in one coherent process here, okay, complementary strands are going to be formed when helicase breaks the hydrogen bonds between the two strands and separates the parent strands. Okay, those free nucleotides are going to kind of attach themselves, okay, to those exposed strands via the rules of complementary base pairing. Again, that involves hydrogen bonds. DNA polymerase is going to take these newly attached strands, or not attached strands, but these new pairs of nucleotides, and it's going to help to form that covalent bond between there. Okay, so that's going to look, well, not like that because that's wrong. That's going to look something, holy cannoli. It's going to look something like this. Okay, so here's what my parent strand, and it's already got these phosphodiester bonds here. Okay, the new nucleotides are going to kind of attach themselves to the exposed strand automatically via the rules of complementary base pairing. Okay, what DNA polymerase is doing is it's going to help form this bond. Okay, so forming this bond between the new nucleotides is kind of the job of DNA polymerase. Now, if we're talking about the human genome, every time that your DNA gets replicated, 3 billion base pairs need to be added, okay? So how is this working? Well, again, the structure of DNA allows for proper copying, okay? So the fact that A can only pair with T, G can only pair with C, okay, means that this process is going to be almost foolproof. And what's going to result are 2 new molecules that are identical to each other, okay? And each one of them contains one of the original strands and one of the newly synthesized strands. But the sequences are exactly the same as the original. Now, how the heck do we know that? Because DNA is too small for us to really take a look at this process, right? So in comes the scientific team of Meselson and Stahl, and this is one of those scientific discoveries that you need to know by name. And they were trying to figure out if DNA replication was conservative or semi-conservative. We're not talking political candidates here, okay? What this means is, was DNA replication a matter of, well, am I producing an entirely new copy, or am I each with one new strand and one old strand, okay? So this is what they were trying to figure out. And the way that they did that was absolutely genius. So they grew a culture of bacteria. Again, they reproduce really quickly, so it's really easy. And they grew them in a broth that had a heavy nitrogen isotope. Okay, and so that heavy nitrogen isotope attached to the DNA. Okay, and because it was heavy, it sank to the bottom of their test tubes. Then they moved those bacteria into a lighter nitrogen isotope. Okay, so they moved it into a medium, causing any DNA replicated in that medium to float farther up into the test tube. It wasn't as dense. Okay, now when they extracted the DNA from the bacteria that was allowed to replicate in the lighter nitrogen, okay, it ended up not at the bottom, but right here in the middle of the test tube, okay? So the blue strand here started out in the heavy nitrogen when it was allowed to reproduce. Then that DNA started to float here in the middle. When they allowed that bacterium to reproduce again in the lighter nitrogen, what they found in their test tubes were two bands. Okay, a band in the middle. Okay, and to them that meant that there was some heavy and some light nitrogen. And they found a band all the way up at the top, which meant that it had to be all light nitrogen isotope. Okay, and what this did is it proved that DNA replication was semi-conservative, and I'll show you what that looks like from top to bottom. So here they are, starting with this heavy DNA, okay, nitrogen 15. When they moved it to the lighter medium, the first thing that's going to happen is those original strands are going to unzip. Again, DNA helicase here. When DNA was allowed to replicate in the lighter nitrogen, okay, that allowed each of these original strands to be used as a template to build a complementary strand. But since now our medium is in a lighter nitrogen isotope, again, we went from where all the DNA sunk down at the bottom because it was heavy to now the DNA is all here in the middle because it's half light and half heavy. When we allowed that to replicate again, what's going to happen is this strand and this strand are both going to be unzipped by helicase. And then new complementary strands, again grown in the lighter nitrogen, are added to each of these. So this is where we're getting some of these in the middle, again, because they're half light and half heavy, and some of them float to the top. And so this proved that DNA was what we call semi-conservative. Okay, they knew that half of the DNA was the original parent strand and half of it was the lighter, newly synthesized DNA. And that is the Meselson-Stahl experiment. You need to know both how they grew the different DNA strands here and how the density of those showed what was going on in the actual process. Okay, so DNA replication is done. We've talked about that, it's semi-conservative. Now we're going to move on to the next two processes, which are transcription and translation. And those are processes that have to do with making proteins. Okay, so who gives a crap about making proteins? Well, proteins actually control all of our cellular processes. So they can act as enzymes, they can be structural, they can help with materials moving in and out of cells, and they're also highly involved in gene expression. Okay, so when I say gene expression, expression of a certain trait or what makes liver cells different from kidney cells. Okay, they express different traits because they make different proteins. Now, DNA has all the codes for making those proteins, but it's lazy. It's the laziest molecule in your whole entire cellular structure, okay? It is stuck here in the nucleus. Of course, here we're talking about eukaryotic cells. Okay, so because it's stuck in the nucleus, we're in a little bit of a jam because we know that proteins are synthesized on ribosomes. So here we have the codes. Here's where we actually do the work. How do we get the information to the workers? Well, to do that, we're going to need some basic steps. Okay, we're going to go into these in more detail later, but it works a little something like this. The codes in DNA are going to be transcribed onto a molecule called RNA. We've already talked about RNA, single-stranded, ribose instead of deoxyribose, U instead of T, etc. The RNA is then going to carry those codes to the ribosome. Then the ribosome is going to translate those codes into a chain of amino acids called a polypeptide. And then those amino acids are going to be folded into their functional shape and travel to their final destination. Now, this process is only going to happen one gene at a time. And remember, a gene is a section of DNA that contains the base sequences necessary to produce one polypeptide. Okay, so if you have a protein like hemoglobin, for example, that's made out of 4 polypeptides, then you would need 4 genes or 4 segments of DNA to carry out all of its processes and make the 4 polypeptides necessary for that. Okay? Now, since DNA can't leave the nucleus to tell the ribosomes what sequence of amino acids need to be put together, we're going to use a molecule called mRNA. This process of manufacturing that mRNA happens in the nucleus, and it's called transcription. I remember that. Script meaning like to write, so here we're writing codes. And that works a little something like this. When we need a particular protein, okay, or polypeptide, the gene or the segment of DNA that contains the codes for that protein is going to get unzipped. Here we're not going to use helicase. We're going to use an enzyme called RNA polymerase, and it's kind of awesome because RNA polymerase does the work of both helicase, like unzipping the DNA, and of making a complementary strand. So it does a little bit of double duty here. So it's going to use one strand of that DNA as a template for making a strand of mRNA. So it reads the codes on that unzipped strand, and then it synthesizes a strand of DNA that has complementary bases. Okay, so it's using complementary base pairing rules to make the strand of mRNA that has those codes. Now, there's a couple things to remember here. Okay, only one parent strand is used as a template because RNA is a single strand molecule, so we only need one strand to act as a template. Let's see. mRNA has the base uracil instead of thymine. So when we're making complementary base pairing, again, G is going to pair C. Okay, if I see a T, I'm going to put an A on the mRNA. But if I see an A, normally T would be complementary, but in this case, it's U. Okay, and of course, the mRNA nucleotides are just a little bit different because they have the sugar ribose instead of deoxyribose. So again, we're unzipping the parent strand. RNA polymerase then uses the exposed strand to make a new strand of complementary base pairs, okay, that are almost exactly the same as the process of making a complementary DNA strand, except we're only using one strand. We have the sugar ribose instead of deoxyribose, and instead of T's, we have U's or uracil. Okay, so let's work on drawing this out. Okay, so I'm going to draw a simplified DNA molecule, meaning I'm not going to draw all of the sugars and phosphates and all that kind of stuff. So I'm going to start off with a segment of that DNA that's still put together. Okay, so let's say that's part of the DNA that I don't need. It's not part of the gene that I need in order to make this polypeptide. Okay, so then I'm going to have a section that's been unzipped. Okay, and this unzipped section reads G, A, C, T, A, G, T. Now, what that means is that in DNA terms, down here would be a C, T, G. So I'm just writing what the complementary base pairs would be on the other strand that's been unzipped. Okay, so there's my DNA. I'm going to build the complementary strand of mRNA in a different color. Okay, so I'm going to do mRNA in red. So here's my strand of mRNA, and it would have the base pair sequence of C, because C is complementary to G, U, so I would normally put a T if I'm building a complementary base pair, but in this case I don't have T's, I have U's. Then it would be G, A, U, U, C, A. Okay, and down here would be any other letters that I've already transcribed. Okay, so this is mRNA. Down here, a lot of people seem to think that U turns into T or T turns into U. That's not how it works. Okay, nothing's turning into anything. These are not copying either. We're not copying the codes. Look, because if I was copying, this would be a G, this would be an A, this would be a C. We're not copying. We're building a complementary strand, okay, using the unzipped parent strand as a template. And of course, all of this is done using the enzyme called RNA polymerase. Now, what you're going to find with the mRNA codes is that we generally look at them in groups of 3, okay? So in DNA, again, so here's the DNA, okay, a gene, a section of DNA. Those groups of 3 base pairs are called triplets. So on DNA, they're called triplets. You have to remember that. On mRNA, we call those groups of 3 codons. Okay, so when we transcribe those codes onto mRNA, and we're writing the complementary base pairs, we're then going to group those base pairs on mRNA into groups of 3 called codons. Now, you should be able to work out some basic math knowing that now, right? So let's say I have an amino acid chain or a polypeptide chain that has 4 amino acids. Since it took 3 codons, sorry, not 3 codons, but since it took 1 codon to make each of those, and each codon has 3 base pairs, that must mean that it took 12 base pairs to make that. Okay, so each amino acid takes 3 base pairs. So if something has 4 amino acids in its chain, it had to have 12 base pair letters, okay, in that original DNA sequence. I can also work the other way around, okay? If I have something that took 300 base pairs to manufacture, that must mean that I could have made 100 amino acids, okay, because 3 base pairs for each amino acid. Now, in the next process, we're going to start talking about some different types of RNA. So let's back up and talk about the different variations of RNA. We know that all RNA is single-stranded, has ribose, has uracil instead of thymine, but there are 3 different variations on RNA. The first one, which you should already be familiar with, is mRNA, or messenger RNA. And again, this serves as the temporary copy of DNA, and it carries those complementary DNA codes from the nucleus to the ribosomes. How's it getting out of the nucleus? Well, remember that the nucleus has these holes called nuclear pores. okay? And it has nuclear pores to get those mRNA molecules out. We also have my favorite, rRNA. Sounds like a seal. Okay, rRNA makes up the ribosome. Okay, so the ribosome itself is made of RNA. And then we have this cool looking dude over here. This is tRNA or transfer RNA, and it transfers amino acids to the ribosomes, okay, to form a growing polypeptide chain. Now, let's take a closer look at this tRNA. Okay, so tRNA doesn't look single-stranded, does it? It looks double-stranded, but don't be fooled. Okay, it's actually still all one single strand that's been folded up on itself due to hydrogen bonds forming between some of those base pairs. Okay, so still all one single strand that's just been folded up. And it's got a couple different parts here. A part where the amino acid attaches, and it's got a part down here called an anticodon. So if I was going to draw an amino, or sorry, not an amino acid, but tRNA, I usually draw it like this. Something like that. And then up here we've got the amino acid. And down here we have 3 base pairs. See how they kind of stick out here? They kind of, instead of folding up and facing the inside, they face the outside. That's strange, huh? And that's called the anticodon. So let's go ahead and annotate here. Again, this is tRNA. And this is the amino acid. And this is called the anticodon. And its function is to bind to a specific mRNA. Sorry, I'm running out of room here. Ugh. Bind to a specific mRNA codon, okay? So it's called the anticodon. It's the exact opposite or complementary sequence of a particular codon. And for each specific anticodon, there's a particular amino acid. And all of this is going to come into play in a process called translation. So again, translation is going to be carried out by the ribosomes, but we know that ribosomes can be found in 2 parts, either attached to the rough endoplasmic reticulum or free-floating out here in the cytoplasm. These free-floating ribosomes are for use when we want to synthesize a protein that's going to stay in the cell. The rough ER ribosomes are going to be making proteins that are then going to be shipped out and packaged into vesicles and secreted from the cell by the Golgi. So where we synthesize a ribosome, where we synthesize a polypeptide or a protein tells us a lot about the function, okay, and where it's going. So let's check out that process, okay? So in the translation process, the mRNA has already left the nucleus through the nuclear pore, and it comes out here and it binds to a ribosome. Now, that ribosome is going to read the codons one at a time. So when I say a codon, I mean a group of 3. So it's going to read these codons, and it's going to bring in a tRNA molecule that has an anticodon that is complementary to the codon that it's reading. So, for example, this next codon in the chain says AAA. What would be complementary to AAA? Well, I know in DNA we would think TTT, but RNA doesn't have T's, it has U's. So in this case, we're looking for UUU anticodon. Okay? Now, that tRNA with its specific anticodon carries a specific amino acid. Okay, so they're all unique. This process is going to keep repeating. Another tRNA with the next proper anticodon in its own amino acid is going to attach to the mRNA molecule. An enzyme is going to attach the amino acids. So we can see that the tRNAs are kind of getting the amino acids in place. We're going to use an enzyme to create these peptide bonds, like to bond them together. Remember, that's a condensation reaction. An anabolic reaction requires energy, okay, probably using an enzyme. And as it's connecting those amino acids, we can start to see a growing polypeptide chain. As that chain is growing, okay, we need this ribosome to keep moving on down this RNA molecule. Once the tRNA has kind of dropped off its amino acid in the chain, we don't need it anymore. So a bond is going to break between the tRNA and the amino acid that it's holding, so it can be ejected from this process. It can get on out of here. Then the ribosome moves down to the next codon, and this process keeps getting repeated. So next in line, these amino acids would join together. This tRNA would get on out of here, and then the ribosome would move down and begin that process on the next one. And what we're going to see is all of these amino acids start to come together in a polypeptide chain. So let's work on drawing this. The first thing that we're going to need is a strand of mRNA. And mRNA, I'm not going to write the codes, okay, has bases on it. And these bases are grouped into threes. And these groups of 3 bases, again, each are called codons. Okay, now a molecule of tRNA with a specific anticodon and its own specific amino acid is going to bind to the codon. Same thing here. In the next one, the next tRNA molecule with its own anticodon and its own amino acid is going to come in here. An enzyme is going to bond these together. Eventually, we're going to have a very long polypeptide chain, okay, a long chain of amino acids, and all of the tRNA molecules that we don't need anymore are getting ejected from this process, okay? All of this happens on the structure known as the ribosome. Okay, so again, here's my tRNA. Okay, and the ribosome is going to be moving this way. Now, let's practice what this looks like kind of in real time. Let's say I have a DNA code that has the following triplets. Okay, GGG, ACT, GCG. Again, those are triplets when they're on DNA. They're going to be transcribed onto a copy of mRNA, and when we transcribe them, they're going to be complementary. So complementary to G would be C. C, C. Yeah, that's a crappy C. Let's do that over again. U, G, A, C, G, C. So there are my mRNA codons. Now that they're on mRNA, we call them codons, not triplets. So the first thing I would want to do is look at my first codon, and then I'm going to reference it in this ultra blurry, super crappy chart. Okay, so here's how we do that. I find the first letter in the codon, which for me is C. So this says first letter. I'm going to be looking in this column. I'm looking for C. My second letter is also a C. So in this row, I find a C. So that must mean I'm looking somewhere in here. My third letter is also a C, so I come here to third letter. I see that it's a C, and that tells me that my first amino acid is proline. Okay, now let's do that for the next one. So the next one is U, so I'm looking somewhere in here. So that's U, then G. So here's the G column. Okay, and then A. So A. Great. Wonderful. I've managed to give you one of the exceptions here. That was really dumb of me. The next amino acid is not an amino acid at all, but it's a stop message. Okay, so I'm not even going to bother translating this one because it just told me to stop. That's the end of my polypeptide. If I was to translate that last one, it would look something like this. So I'd be looking at C, so somewhere in this row, G, so somewhere in this row, and then C, right here, so arginine. But I don't need to do that because it already told me to stop. Next thing I want to look at is how we can kind of use this chart to figure out what code I would need to make a particular amino acid. So if I wanted to produce isoleucine, okay, I need to find isoleucine in my chart here. And I see it right here and right here. So it's looking to me like I can have two different codons to make this. A, U, C, and A, U, A, both make isoleucine. So in some cases, we can have multiple options or multiple ways of getting the same amino acid. Now, one of the things that's really cool about your genetic code is that it is universal. And so what that means is that no matter what organism you're looking at, okay, the DNA works the same way. The same DNA code in shrimp gets transcribed into the same mRNA code, okay, in butterflies. It doesn't matter, okay? A is always complementary to T, no matter what organism you're looking at. And these get translated into the same amino acid, okay, in every organism. So it works the same way in every organism. That DNA code is universal. You don't need to go looking for one of these charts for humans and a different chart for cows and a different chart for bacteria. It all works the same. And that allows us to do some wicked cool things. Okay, so one of the things that we're able to do is insert genes from different species into the genomes of entirely different organisms. Okay, we call that gene transfer. We're going to talk about that later on in another chapter. But one example that I think you'll be able to understand now is making insulin. So way back in the day, if someone was diabetic and they needed insulin, what they had to do is literally harvest insulin from the pancreas of pigs. And that was really expensive. Imagine having to grow a whole pig only to get a little bit of insulin out. Well, now what we've done is we've isolated the human gene for insulin, and we can take that gene and glue it into the DNA sequence of bacteria. We chose bacteria because their genome is simple and because they can grow and reproduce really quickly. So once that gene is inserted into here, those codes get transcribed and translated just the same as it would if it were in a human. These A's, T's, G's, and C's mean the same thing in humans as they do in bacteria. And so what that does is it codes for the same polypeptide or the same amino acid sequence, and we get an identical protein. Okay, insulin is a protein, so we're able to then just harvest that amino acid sequence, that protein, and it is exactly the same coming from this process as it would be if it were naturally occurring in humans. So something really cool there. So just a quick review of transcription and translation. They're very easy to get confused, hence why they're a very common question on your IB exams. Okay, so you'll get things with such command terms like compare transcription and translation, distinguish between transcription and translation, etc., right? So transcription again occurs in the nucleus, and I know that because I'm using DNA as a template to make mRNA. So in eukaryotes, if I'm using DNA, I gotta be hanging out in the nucleus. In translation, that's going to occur in the cytoplasm on one of the ribosomes, and its final result is a growing polypeptide chain. So I keep reiterating the importance of enzymes in all of these processes, okay? Now, one of the processes, again, that we're going to talk about in multiple different chapters is this process called PCR, and PCR stands for Polymerase Chain Reaction. Hold on, wait a second. We've heard of this polymerase before. That's the enzyme that's used to copy DNA, okay, to make a complementary strand during DNA replication. Well, we've been able to use this process to our advantage for such things as forensics. So let's say you're investigating a crime scene and you find out that the killer has left a tiny hair or a cell or something, and you want to be able to use that DNA to identify that suspect. Well, in order to do that, you're going to need a crap load of copies of that DNA. And so to make all those copies, we use, again, polymerase chain reaction. PCR works something like this. Okay, first we have to separate the two parent strands. So here they are, they're already together. We have to separate them. In real human cells, that would be done using DNA helicase, but in this case, we're just going to use heat. One of the things we know about heat is it breaks hydrogen bonds. That's why heat is able to denature proteins. It ruins the hydrogen bonds that hold them in place. So we use heat to separate the two strands. The single strands are added to solutions that are full of free nucleotides, okay, and an enzyme called polymerase. The enzyme, okay, kind of helps these free nucleotides to form complementary strands. And this happens over and over and over again, okay? The problem is that we know enzymes are susceptible to heat. So how do we deal with that? We need the heat to break the hydrogen bonds, but we don't want it to be so hot that it denatures that DNA polymerase. If we were using human DNA polymerase, the heat necessary to separate the two strands would denature that enzyme. So we found a polymerase, a DNA polymerase, so it works the exact same way, but we found it in another organism. We found these really cool heat-resistant bacteria. They're called thermophiles. They live in these really hot areas, and so their enzymes have different optimal temperatures. So whereas human DNA polymerase, well, that's not right. Human DNA polymerase has kind of something like this, okay, an optimal temperature here. Taq polymerase has an optimal temperature all the way up here. So it's optimal and working fastest at a much different temperature range. Okay, we call it Taq, okay, from T. aquaticus. That's where we get that name from. Okay, and so it allows us to be able to use a polymerase enzyme without having to worry about denaturing it. So we need that heat to separate these two strands, but we also need an enzyme to help facilitate the building of the complementary strands. We were able to find the same enzyme with a different optimal temperature that works for our process. So kind of cool. Again, understanding that you're never ever going to get away from enzymes in IB Bio. And that'll do it for chapter 2.7.